Hello students, today I bring for you a new chapter, that is photosynthesis in higher plants. In this chapter we are going to cover many important topics, so let's start. First of all, basic photosynthesis. So photosynthesis is a physico-chemical process by which green plants use light energy to synthesize organic compounds. You remember the term physico-chemical process, that is also mentioned in your NCERT, and many questions are coming from this word. Okay, it is the basis of life on earth. Ultimately all living forms depends on sunlight for energy. Now, importance of photosynthesis: it is the primary source of all food on earth. It releases oxygen into the atmosphere. Now some experiments related with photosynthesis. Some experiments we have done in class 10th in chapter life process, and now just a reminder and then start the new experiment which are actually on your NCERT. First of all, variegated leaf experiment. Take a variegated leaf or a leaf partially covered with black paper that was exposed to light. Test the leaf for starch. It shows that photosynthesis occurs only in green part of the leaf in presence of light. Second is half leaf experiment, that is, a part of leaf is enclosed in a test tube containing KOH soaked cotton which absorbs CO2. The other half of the leaf is exposed to air. Place the setup in light for some time. Test the leaf for presence of starch. Exposed part shows positive for starch and portion in the tube shows negative. This proves that CO2 is required for photosynthesis. These two experiments are in your class 10th NCERT chapter life process. Next, these experiments are on your 11th biology NCERT. First of all, experiments by Joseph Priestley, 1770. Priestley performed experiments to prove the role of air in the growth of green plants. He discovered oxygen in 1774. He observed that a candle burning in a closed bell jar gets extinguished. Similarly, a mouse suffocated in closed jar. He concluded that a burning candle or a breathing animal damages the air. He placed a mint plant in the bell jar, as you see in this diagram. He found that the mouse stayed alive and the candle continues to burn. He hypothesized that plants restore to the air whatever breathing animals and burning candle remove. Next experiment by John Ingenhousz, 1732-1799. He conducted the same experiment by placing it once in the dark and once in the sunlight. He showed that sunlight is essential to the plant for purifying the air. He repeated this experiment with an aquatic plant. It shows that in bright sunlight, small bubbles are formed around green part while in the dark they did not. Later he identified these bubbles to be of oxygen. Thus he showed that only the green part of the plants release oxygen. You can see in this diagram how the air bubbles are formed in this plant. Next experiment by Julius von Sachs, 1854. He proved that glucose is produced when plants grow and it is usually stored as starch. Chlorophyll is located in special bodies that are called chloroplast. Glucose is made in the green part of the plant. Next important experiment by T.W. Engelmann, 1843 to 1909. What he did now, he split the light using a prism into its spectral components and illuminated a green alga, that is cladophora, placed in a suspension of aerobic bacteria. The bacteria were used to detect the site of oxygen evolution. He observed that the bacteria accumulated mainly in the region of blue and red light of the split spectrum. It was the first described action spectrum of photosynthesis. It resembles the absorption of spectra of chlorophyll A and B. Like this diagram, you can see light goes into the prism and it then split out and becomes seven different colors, as you all know, Vivgor. And you can see this is the particular cladophora, this portion. Okay, it is cladophora. Okay, and some aerotactic bacteria or aerobic bacteria are present like this. So when these bacteria, so what he concluded, now mainly the bacteria accommodated mainly in the region of blue and red light. Like this region is the blue region, as you can see, and this one is the red one. Okay, so he concluded he first described action spectrum of the photosynthesis. Next, by the middle of 19th century, it is discovered that plants use light energy to make carbohydrates from CO2 and H2O. Empirical equation of the process of photosynthesis is CO2 plus H2O gives rise to CH2O plus O2. Empirical means that equation that accepted by all over the world. Okay, empirical term, where CH2O represents a carbohydrate, example glucose. Next experiment by Cornelius von Neil. Cornelius von Neil, 1897 to 1985. Von Neil, a microbiologist, conducted some studies in purple and green bacteria. He demonstrated that photosynthesis is a light-dependent reaction in which hydrogen from a suitable oxidizable compound reduces CO2 to carbohydrate. This can be expressed by 2H2A plus CO2 gives rise to 2A plus CH2O plus H2O. So, in plant, H2O is the hydrogen donor and is oxidized to O2. Purple and green sulfur bacteria use H2S as hydrogen donor. So, the oxidation product is sulfur or sulfate and no oxygen is produced. Thus, he inferred that the O2 evolved by green plant comes from H2O, not from CO2. This was later proved by using radioisotopic techniques. Therefore, overall correct equation for photosynthesis is 6CO2 plus 12H2O gives rise to C6H12O6 plus 6H2O plus 6O2. Okay. Remember the scientist's name and what he did in his experiment. What he concluded. So here Van Neil concluded that oxygen evolved by green plants. Oxygen evolved by green plant comes from H2O, not from CO2. Okay. It was demonstrated by Van Neil. Direct question comes. Okay. Remember the term. Now, photosynthesis, sites and pigments. First of all, chloroplast. We all know that plant have a particular plastid that makes the process of the photosynthesis. So, chloroplast is here. Diagram also in your NCERT as in your screen also. Photosynthesis takes place in green leaves and other green parts of the plant. Chloroplast present in the walls of mesophyll cells of leaves. It helps to get optimum quantity of incident light, means direct sunlight. Chloroplast contains a membranous system. It consists of grana, stroma lamellae and fluid stroma. Each granum is a group of membrane-bound sac called thylakoids or lamellae. They contain leaf pigment. The membrane system traps light energy and synthesis ATP and NADPH, as you can see in this diagram. It is called light reaction. In stroma, enzymatic reactions incorporate CO2 into the plant for synthesizing sugar, which in turn forms starch. It is called dark reactions or also called carbon reaction. Okay, it does not mean that they occur in darkness or that they are not light dependent. Remember, dark reaction means it does not occur in the darkness only or they are not light dependent. Okay. Now some pigments that involve in the photosynthesis. Pigments are substances that have ability to absorb light at specific wavelengths. Chromatography shows that following leaf pigments in chromatogram. Chlorophyll A, bright or blue-green, as you can see in this diagram. Chlorophyll B, yellow-green, xanthophylls, yellow, carotenoids, yellow to yellow-orange. This is the color that shows by chromatogram. Okay, how this the, so there is a practical about the chromatography. You all did do this practical in your biology lab. Okay, to separate the pigments of plant. And finally, what the final output comes? Now in a paper, you easily identified chlorophyll B, chlorophyll A, xanthophylls and carotenoids. Next, chlorophyll B, xanthophylls and carotenoids are accessory pigments. Remember the term, chlorophyll B, xanthophylls and carotenoids are accessory pigments. Function of accessory pigments, now they absorb light at different wavelengths and transfer the energy to chlorophyll A. They protect chlorophyll A from photooxidation. Now, the absorption spectrum and action spectrum coincide closely, showing that photosynthesis is maximum at blue and red region of the spectrum. The graph also shows that chlorophyll A is the chief pigment associated with the photosynthesis. You can see 3 diagrams are also in your NCERT, and here the chlorophyll A is the chief pigment while the others are associated. Now, photosystems. Pigments are organized into 2 photosystems called photosystem 1 or PS1 and photosystem 2 or PS2. They are named in the sequence of their discovery. Each photosystem has a chlorophyll A and accessory pigments bound by proteins. All pigments except one molecule of chlorophyll A form a light harvesting complex LHC. All pigments except one means you can see these are the pigment molecules. Okay, all are form a light harvesting complex, and this is the light harvesting complex. Okay, except one chlorophyll A, that is this one. Okay, except this one. Single chlorophyll A acts as reaction center. It is also labeled here. You can see single chlorophyll A act as reaction center. In PS1, the reaction center absorbs light at 700 nanometer and so called P700. In PS2, the reaction center absorbs light energy at 680 nm and so called P680. Next, light reaction or photochemical phases. Light reaction includes light absorption, water splitting, oxygen release and the formation of ATP and NADPH. See this diagram? Light absorbed in the leaf, water split out and oxygen release and formation of ATP and NADPH. ATP and NADPH are high energy chemical intermediates. The electron transport. When PS2 absorbs red light of 680 nm wavelength, electrons are excited and transferred to an electron acceptor. Here you can see light energy comes to the LHC, and where the electrons are excited, transferred to the electron acceptor like this. Okay. The electron acceptor passes them to a chain of electron transport system. So here there is chain of electron transport system consisting of cytochromes. We will discuss about this later. This movement of electrons is downhill in terms of redox potential scale. The electrons are transferred to the pigment of PS1. Then the electron transferred to the pigments of the PS1 light harvesting complex. Next, simultaneously, electrons in PS1 are also excited when they receive red light of 700 nanometer and are transferred to another acceptor molecule having a greater redox potential. So when the 700 nanometer light comes, PS1 are excited, electrons are excited, and they transfer to the another electron acceptor. Okay. These electrons are moved downhill to a molecule of NADP plus. Then they move to downhill to NADP plus, and as a result NADP plus is reduced to NADPH and H plus. Transfer of electrons from PS2 to PS1 and finally downhill to NADP plus is called the Z scheme. Here you can see first goes to this one, then electron transport system, then goes to this, and then comes to NADP plus. That's why it is called Z scheme due to its zigzag shape. This shape is formed when all the carriers are placed in a sequence on a redox potential scale. Next is photolysis or splitting of water. The water splitting complex in PS2 is located on the inner side of the thylakoid membrane. Water split into H plus, O and electrons. 2H2O gives rise to 4H plus O2 plus 4 electrons. So PS2 can supply electrons continuously by replacing electrons from water splitting. Thus PS2 provides electrons needed to replace those removed from PS1. The proton H plus are used to reduce NADP to NADPH, and oxygen is liberated as a byproduct of photosynthesis. Next is photophosphorylation. The synthesis of ATP by cell in mitochondria and chloroplast is called phosphorylation. Photophosphorylation is the synthesis of ATP from ADP in chloroplast in presence of light. It occurs in two ways, non-cyclic and cyclic. First of all, non-cyclic photophosphorylation. It occurs when the two photosystems work in a series, first PS2 and then PS1, through an electron transport chain as seen in the Z scheme. Here ATP and NADPH and plus H plus are synthesized. It is a non-cyclic process because the electrons lost by PS2 do not come back to it but pass on to NADP. Why it is called non-cyclic? Because the electrons that are from PS2 to PS1, they did not move back. Okay, they do not move back because they goes to NADP plus. Okay, that's why it is called non-cyclic. Next is cyclic photophosphorylation. It occurs in only photosystem 1. So it occurs in the stroma lamellae when only PS1 is functional. The electrons is circulated within the photosystem and ATP synthesis occurs due to cyclic flow of electrons. The lamellae of grana have PS1 and PS2, but the stroma lamellae lack PS2 and NADP reductase. The electron does not pass on to the NADP plus but is cycled back to PS1 through electron transport chain. Here only ATP synthesized, no NADPH or H plus. As you can see, only ATP synthesized. Cyclic photophosphorylation also occurs when only light of wavelength beyond 680 nanometer are available. Now, chemiosmotic hypothesis. It explains mechanism of ATP synthesis in chloroplast. Chemiosmosis means movement of ions across a semi-permeable membrane. It occurs in chloroplast and mitochondria. Chemiosmosis needs a membrane, a proton pump, a proton gradient across thylakoid membranes and ATP synthesis. Splitting of water takes place on the inner side of the membrane, so the protons accumulated in the lumen of thylakoid. As an electron moves through photosystem, protons are transported across the membrane. It is due to the removal of protons from stroma for the following reason. First one, the primary electron acceptor is located towards the outer side of the membrane and transfers its electron to an H carrier, hydrogen carrier. So this molecule removes a proton from the stroma while transporting an electron. When this molecule passes on its electron to the electron carrier on the inner side of the membrane, proton is released into the lumen of the membrane. Second, The NADP reductase enzyme is located on the stroma side of the membrane along with electron coming from PS1. Protons are necessary to reduce NADP+. These protons are also removed from stroma. Hence, protons in stroma are decreased, but in lumen, protons are accumulated. So, as you can see, lumen having high H plus ion, that are protons. It creates a proton gradient across the thylakoid membrane and decrease in the pH in the lumen. Breakdown of proton gradient leads to synthesis of ATP by ATP synthase enzyme. As you can see, this is PS2, light comes to PS2, then electrons, then it goes to plastoquinone, then cytochrome B6F, then plastocyanin, then comes to PS1. Okay, here FD and FNR are mentioned. FD means ferredoxin, FNR means ferredoxin NADP reductase. Then it goes to NADP, then NADPH+. ATP synthesis consists of two parts. One is the CF0 and CF1. CF0, you can see it is embedded in the membrane. As you can see, CF0 is embedded in the membrane and forms a transmembrane channel. It carries out facilitated diffusion of protons across the membrane to the stroma. It results in the breakdown of proton gradient. CF1, it protrudes on the outer surface of the thylakoid membrane. The energy due to breakdown of gradient causes a conformational changes in the CF1 particles. It makes the enzyme to synthesize ATP. Energy is used to pump protons across a membrane to create a gradient or a high concentration of protons within the thylakoid lumen. ATP synthesis has a channel for the diffusion of protons back across the membrane. This releases energy to activate ATP synthesis that catalyzes formation of ATP. Now, dark reaction or biosynthetic phase. Use of the ATP and NADPH. First of all, product of light reactions are ATP, NADPH and O2. Dark reaction is the use of ATP and NADPH to drive the process for the synthesis of foods, that are sugars. This phase does not directly depend on the light but is dependent on the product of the light reaction. It can be verified as follows. Immediately after light becomes unavailable, the biosynthetic process continues for some time and then stops. If light is available, the synthesis starts again. Here you can see CO2 combines with H2O to form CH2O whole N or sugars. CO2 assimilation during photosynthesis is two types, C3 pathway and C4 pathway. First of all, C3 pathway. In this, first product of CO2 fixation is the C3 organic acid, that is also called 3-phosphoglyceric acid or 3-PGA. Melvin Calvin discovered this using a radioactive C14 in algal photosynthesis. C4 pathway. In this, first table product is oxaloacetic acid OAA, a 4-carbon C4 acid. First of all, C3 pathway or Calvin cycle. It occurs in all photosynthetic plants, C3 or C4 pathway. It has 3 stages: carboxylation, reduction and regeneration. First of all, carboxylation of RUBP. What is RUBP? Now RUBP is a ribulose bisphosphate or a 5-carbon ketone sugar. It is the primary CO2 acceptor who receives the CO2. It is the most crucial step. CO2 is fixed by RUBP to 2,3-PGA in presence of enzyme RUBP carboxylase. Since this enzyme also has an oxygenation activity, it is called RUBP carboxylase oxygenase, or in shortcut it is called Rubisco. Rubisco is the most abundant enzyme in the world. Second is reduction. It is a series of reactions leading to the glucose formation. Here 2 ATP molecules for phosphorylation and 2 NADPH for reduction per CO2 molecules are used. Fixation of 6 CO2 molecules and 6 turns of the cycle are needed to remove one glucose molecule from the pathway. Next is your regeneration. It is a crucial for continuation of the cycle. It requires 1 ATP for phosphorylation to form RUBP. Hence, for every CO2 molecule, 3 ATP molecules and 2 NADPH are required. It is probably to meet this difference in number of ATP and NADPH usually in the dark reaction that the cyclic phosphorylation takes place. To make one glucose of molecule, 6 turns of the cycles are needed. So here what goes in and comes out of the Calvin cycle. Here you can see 6 CO2 goes in and 1 glucose comes out. 18 ATP goes in and 18 ADPR comes out. 12 NADPH goes in and 12 NADPR comes out. Now C4 pathway or Hatch and Slack pathway. It is present in the plant adapted to dry tropical regions. They also use C3 pathway as main biosynthetic pathway. The large cells around the vascular bundles of the C4 plant are called bundle cell. See, it is also labeled. Such anatomy or such arrangement is called Cranch anatomy. The bundle cells may form several layers around the vascular bundle. They have large number of chloroplasts, thick walls, impervious to gas exchange, and no intercellular spaces. Cranch means rats. Steps in the Hatch and Slack pathway. Primary CO2 acceptor is phosphoryl pyruvate, PEP also called. It is a 3-carbon molecule seen in the mesophyll cells. As you can see, this is the mesophyll cell labeled here, and atmospheric CO2 absorbed by phosphoryl pyruvate. Okay. The enzyme for the fixation is PEP carboxylase or PEPkase. Which enzyme is used to trap the atmospheric CO2 into the phosphoryl pyruvate? Now, PEP carboxylase or PEPkase. The mesophyll cells lack Rubisco enzyme. Mesophyll cells do not have Rubisco enzyme. The C4 acid OAA is formed in mesophyll cells. As you can see, C4 acid is labeled. C4 acid means that is oxaloacetic acid. It then forms other 4 carbons like malic acid and aspartic acid. Then they are transported to bundle cell. Directly oxaloacetic acid does not come in the bundle cell and does not transport between in the plasmodesmata. So it have to convert to malic acid or aspartic acid. Then they transport it to the bundle cell. Next, in bundle cells, C4 acids are broken down to release CO2 and a C3 molecule. C4 acid is broken down by the process of decarboxylation and CO2 released, as well as C3 acids are produced. The C3 molecule is transported back to the mesophyll cells where it converted to PEP again. So again, the C3 acid transported to the mesophyll cells and converted into PEP again. It again regenerated to convert into phosphoryl pyruvate. Bundle cells are rich in Rubisco but lack PEPkase. Bundle cells are rich in Rubisco but they do not have PEPkase. The C3 pathway is common to C3 and C4 plants. Next, C4 plants are special because they have a special type of leaf anatomy that is called Cranch anatomy. They tolerate higher temperatures. They show a response to high light intensities. They lack photorespiration and they have greater productivity of biomass. Now, in Calvin pathway, RUBP combines with CO2. RUBP plus CO2 in presence of Rubisco 2 into 3 phosphoglyceric acid. Active site of Rubisco can bind to CO2 and O2, hence the name. Rubisco has greater affinity for CO2 than for O2. This binding is competitive. Relative concentration of O2 and CO2 determines which one will bind to the enzyme. In C3 plants, some oxygen binds to Rubisco, hence CO2 friction is decreased. Here RUBP binds with O2 to form one molecule of phosphoglycerate and phosphoglycolate. This pathway is called photorespiration. As you can see, phosphoglycolate and phosphoglycerate are formed. In this pathway, there is no synthesis of sugar, ATP and NADPH. Hence, photorespiration is a wasteful process. Rather, it causes the release of CO2 by using ATP. In C4 plant, photorespiration does not occur because they can increase CO2 concentration at the enzyme site. This takes place when C4 acid from the mesophyll is broken down into the bundle cell to release CO2. This minimizes the oxygen activity of Rubisco. Due to the lack of photorespiration, productivity and yields are better in C4 plants. In addition, these plants show tolerance to higher temperatures. Next, your table is there. You can see C3 and C4 plant. C3 plant photosynthesis occurs in mesophyll cell, but in C4 plant it occurs in mesophyll and bundle cell. C3 plant crans anatomy absent, while in C4 plant it is present. RUBP is the primary CO2 acceptor, and here in C4 plant PEP is the primary CO2 acceptor. 3 PGA and 3 carbon compounds is the first double product, and here oxaloacetic acid 4 carbon compound is the first double product. Chloroplasts are of only one type, granule, but here chloroplasts are dimorphic, granule in mesophyll and agranule in bundle cells. Photorespiratory loss is high in C3 plant, while in C4 plant photorespiration is absent or negligible. High CO2 compensation point 25 to 100 ml of CO2 per liter, while in C4 plant low CO2 compensation point 0 to 10 ml carbon dioxide per liter. Optimum temperature for photosynthesis is about 25 degrees Celsius, while in C4 plant it is about 35 to 45 degrees Celsius. Photosynthetically less efficient and productivity is low in C3 plant, example rice, wheat, bean, potato, but in C4 plant photosynthetically more efficient and productivity is high, example maize, sugarcane, amaranth and sorghum. Now last topic, factors affecting photosynthesis. Here there are some internal as well as external factors are affecting photosynthesis. First of all, internal: number, size, age and orientation of leaf, mesophyll cells and chloroplast. Internal: CO2 concentration and amount of chlorophyll. And external factors are sunlight, temperature, CO2 concentration, water. Plant factors are dependent on genetic predisposition and the growth of the plant. Here, Blackman's law of limiting factor, 1905. If a biochemical process is affected by more than one factor, its rate is determined by the factor nearest to its minimal value. It is the factor which directly affects the process if its quantity is changed. For example, if a plant with a green leaf, optimal light and CO2 may not photosynthesize in very low temperature. If the optimal temperature is given, it will start photosynthesis. So if any one factor is limited, okay, then it directly affects the whole process of photosynthesis. Light also, light quality, light intensity and duration of exposure to light influence photosynthesis. This is a linear relationship between incident light and CO2 fixation rate at low light intensities. At higher light intensities, the rate does not further increase because other factors become limiting. Light saturation occurs at 10% of the full sunlight. Hence, except for the plant in shade or in dense forest, light is rarely a limiting factor in nature. High increase in incident light breaks down chlorophyll, it decreases photosynthesis. As you can see, light goes down and here the rate of photosynthesis. Light comes in and the row goes down, then at a time it becomes parallel to. Why? Because other factors are also be available like this. If more light coming, but it require water also, Okay, that's why it then comes into a parallel line. Next, carbon dioxide concentration, major limiting factor for photosynthesis. CO2 concentration is very low in atmosphere, that is 0.03 to 0.04%. Increase up to 0.05% causes increase in CO2 fixation rate. Beyond this level, it becomes damaging over longer period. At low light conditions, C3 and C4 plants do not respond to high CO2 conditions. At high light intensities, they show increased rate of photosynthesis. C4 plants show saturation about 360 ml per liter. C4 plant respond to increased CO2 concentration and saturation is seen only beyond 450 ml per liter. Thus, current availability of CO2 level is limiting to the C3 plants. Due to response to higher CO2 concentration, C3 plants show increased photosynthesis and higher productivity. This fact is used for some greenhouse crop like tomato, bell peppers, etc. They are grown in CO2 enriched atmosphere that leads to higher yield. Next is your temperature. Dark reactions being enzymatic are temperature controlled. Influence of temperature or light reactions is very less. The C4 plant responds to higher temperatures and so higher rate of photosynthesis. C3 plant have a much lower temperature optimum. The temperature optimum of plants also dependent on their habitat. Tropical plants have higher temperature optimum than the plant adapted to temperate climates. Next finally factor is the water. Water stress closes the stomata, hence reduces the CO2 availability. Water stress also wilts leaves, thus reduce the surface area of the leaf and their metabolic activities. So these are the factors affecting photosynthesis, and with this your important chapter is completed. Read your NCERT by watching this video line by line, underline important terms, and if you face any doubt, simply ask me. My contact number is in your video description. Okay, so like this video, share it with your friends, and if you are new to my video, then subscribe my channel. Thank you. Jai Jagannath.